Hello, AP Bio. Welcome to our video lecture for Chapter 35, the immune system. So the last couple chapters, I've begun with saying that there's two big themes I want you to consider in this set of chapters. One is how cells communicate with one another, and the other is how cells or organisms respond to changes. And the immune system is a terrific example of both of those. So I picked this picture. This is Jack. He's about 2, and he's obviously visiting Santa. And I love this picture, one, because it's just adorable. But he's kind of given the side eye with the Santa, and I don't think he's fallen for it. I don't think he's buying that this is the real Santa Claus. I think he realizes that someone's trying to trick him, and he is skeptical. And the immune system is kind of like that. Your immune system helps your body to determine what should be there, what's really your cells, and what should not be there. And it attacks things that should not be there. And it's very hard to trick your immune system, just like it's very hard to trick 2-year-old Jack as to where the real Santa Claus is, because he's not convinced that this was him. So this chapter, you probably don't have a lot of experience with the immune system, at least in science class. Usually you don't really get to the immune system until AP Bio, or maybe even into college if you take biology in college. So there's lots of vocabulary. So pathogens are just anything that causes you to get sick. So viruses, bacteria, some protists can make you sick. Your immune system is the system that you have that helps limit infections. And there's 2 sort of overall categories of your immune system. There's what's called innate immunity, which is non-specific. Things like your skin. Your skin, it keeps things out that should be out, but it's not specific to certain pathogens. And then you have adaptive immunity, which is specific, say, to the flu or to a cold or to chickenpox. The first one we're going to go through, this is a picture. The green are bacteria, and this big blobble thing is a white blood cell that's engulfing the bacteria, and those bacteria are toast. They are about to be dead. So this diagram is from the book. This goes through the 2 parts of your immune system, innate and adaptive, and basically this sort of summarizes the entire chapter. So you might want to refer to this diagram again at the end. So we're going to start with the innate immunity. These are the non-specific defenses, and these honestly are pretty simple. So the first line of defense are your barrier defenses, things like your skin. The surface of your skin has a decently low pH, maybe around 4 or 5 due to sweat, and that, pathogens don't like it acidic normally. Your saliva, your tears, your mucus traps pathogens. Your trachea is lined with cilia that can help sweep out pathogens also. There's an enzyme called lysozyme. We mentioned this in a previous chapter. It's found in tears, saliva, sweat, and breast milk. And lysozyme helps to break down bacterial cell walls. So your first line of defense, which is actually very, very, very effective, are things that just block out or in different ways disrupt pathogens from entering your body. So let's say that a pathogen does enter your body through maybe a cut. So the second line are called cellular defenses. So phagocytosis is the ingestion of pathogens, and different kinds of white blood cells. White blood cells, again, are cells of your immune system, have these receptors called toll-like receptors, the little sort of odd baseball bat shaped things, like a cartoon baseball bat. These are receptors that are on phagocytotic cells of white blood cells, and basically they will attach to proteins like flagellin. Flagellin is a protein that bacteria have in their flagella. They'll attach to it, and then whatever, this would be attached to a bacteria. Then you're going to get an immune response where the bacteria or the white blood cell engulfs the bacteria and breaks it down. Just to remind you, phagocytosis in this picture kind of goes back to the first picture, where I have a white blood cell, in this case, engulfing something, bringing it in, forming a vacuole or vesicle is probably a better term. And then a lysosome comes and fuses, spills in its acidic contents, its enzymes, breaks it down, and the pathogen is destroyed. This is still part of your innate immunity. This is still non-specific. It might seem kind of specific here, but it's not specific to a certain kind of pathogen, like a strain of the flu, so it's still non-specific. So the second line of defense also includes a series of different types of phagocytotic cells. So the textbook goes through lots of details on all of these. I just want you to be familiar with these 4. Basically, these are 4 different types of white blood cells. You should know, what they are and what they do. So neutrophils are kind of white blood cell Cells that are infected with bacteria or viruses sometimes give off chemicals. They're like a warning sign that someone should probably come kill me because I'm infected. Neutrophils respond to those signals. Macrophages are amoeboid cells that migrate throughout the body and will engulf things that are foreign. Eosinophils are white blood cells that contain destructive enzymes that will break down pathogens. Natural killer cells are white blood cells that will destroy the body's own abnormal cells. Things that might have a viral infection or even cells that are cancerous can be killed by your immune system, by your own cells. If you want to know more details about these, look it up in the book. Another part of the second line of defense, so antimicrobial proteins. So interferons are chemicals that are secreted by infected cells, like we mentioned on the previous slide, that are a warning sign to other cells that, hey, I'm infected, someone should probably come take care of me. The complement system is a system of about 30 proteins that if there's a pathogen, they get activated and they will lyse or break down invading microbes. Another part of the second line of defense is called the inflammatory response, and this is actually pretty important. So this is triggered by physical injury. So you have cells called mast cells that release a chemical called histamine. You've probably heard of antihistamines, medicines you might take if you have allergies. So histamine is a chemical that causes blood vessels to dilate, and the word is vasodilate, remember? And they also become more permeable. So remember, your blood is what carries your immune system largely. So if I have more blood flow to an area, you're going to get a larger immune response, which is an inflammatory response. If you look at this diagram here, I have a wound. Here I have a splinter. There's bacteria on the splinter, so this is super bad. The mast cells secrete histamine, which causes the blood cell to dilate. It becomes more permeable. So these kind of, what are these, either neutrophils, actually leave your blood and go into the surrounding tissues and take care of the pathogen. An antihistamine, like in Sudafed, blocks histamine. So an allergic response might include, like if you have a rash or if you have, some kind of allergy problems, that might be the body reacting, say, to pollen, which pollen, isn't a pathogen. Pollen doesn't, make you sick unless your body responds to pollen. It's almost like your body being overreactive to something that it shouldn't react to at all. So an antihistamine would block histamine, which would prevent this inflammatory response, say, to pollen. A fever, you know what a fever is. Fevers are induced by a substance or substances called pyrogens. They increase your body's thermostat to fight invaders. So this is an interesting point. An infection doesn't cause a fever. You cause the fever, all right? A fever is your body's response to try and fight a pathogen. And fevers really aren't that effective at fighting diseases. It's one of these things your body does that it isn't that effective. But just keep in mind that, like, the symptom of a fever, like a cold itself doesn't cause the fever. Your body causes the fever to fight the cold. Okay, so that's all we're going to say about the innate immune response. Let's just go back for a second to this diagram, this sort of peach or orangey color. These are things that are non-specific that will react to pretty much any kind of pathogen. Very, very effective. A lot of stuff gets kept out of your body by things like your skin. Now, the second system is called adaptive immunity. Let me go back. So this is where it gets kind of complicated. So adaptive immunity, the key is that it adapts and it's specific to very specific pathogens, like this variant of the flu or that type of bacteria. And it relies upon two types of lymphocytes. The term white blood cell, so leukocytes, I don't think I said this, leukocytes is the word for white blood cells. Leuko means white. Lymphocytes are a type of white blood cell. So like neutrophils, eosinophils, natural killer cells, T cells, B cells, they're all leukocytes. Lymphocytes are a type of white blood cell. There's two types. There's T cells and B cells. T cells are made in the thymus gland, which is above your heart. B cells are made, or mature rather, in your bone marrow. And if you look at these two pictures, this is a super important concept to have in your head. So B cells have these Y-shaped surface, membrane surface proteins. T cells have these kind of rod-shaped or maybe eye-shaped. These are called antigen receptors. So antigens are anything that's foreign in your body is an antigen. An antigen receptor is some kind of surface protein or glycoprotein or glycolipid or whatever, some kind of surface feature on a B cell or a T cell that will attach to antigens. Just to make sure we have this clear, think about this question. What makes antigen receptors? You make antigen receptors, all right? Do you make antigens? No, you respond to antigens. People get these terms confused, and actually the next slide, I'll go ahead and put the slide, defines it. Antigens are any substance in your body that elicits an immune response. So anything that's foreign. Although in some autoimmune diseases, like in some kinds of arthritis, your body's attacking the cartilage between your joints, and that's not foreign. But that's your body not doing what it's supposed to be doing. It's kind of going overboard. Antigen receptors are the surface features on B cells or T cells that bind to antigens. Now, this is important. Your immune system can produce millions of different antigen receptors. It's very, very adaptive. But on a single B cell or a single T cell, all the receptors are the same. So if this is a B cell, it might have 100 receptors on it, but they're all identical to the same antigen. So basically, your body can produce millions of different types of B cells and T cells. The word epitope, so the part of the antigen that actually binds to the antigen receptor, like if the antigen could be big, maybe the receptor only binds to one little small part of it. That's called the epitope. And go ahead and keep in mind here, the way this is working, this is a physical binding of the antigen receptor to the antigen, all right? It's a touch. It's two molecules that are similar in shape or whatever, and they actually bind together. If the immune system cannot see the antigen, if it can't actually touch it and bind to it, it can't fight it off, all right? So this gets a little thick, so let's just go through this carefully. So first we're going to discuss B cells and what are called antibodies. So B cells, which are made in your bone marrow, they have these Y-shaped antigen receptors. So this is a little more detailed than we need to get into, but let's just briefly address it. So each of these antigen receptors has a constant region and a variable region. Notice the variable region, which means they can change, is on the end where they bind to the antigen. The constant regions are what binds it to the cell. And what's unique here is that a cell can make millions of different types of receptors by switching out the V parts and the C regions, almost like switching out Legos. So it doesn't take a million different genes to make a million different antigen receptors because you can swap them out, all right? And just note that the variable regions are on the ends where you actually bind to the antigen, okay? So the way that a B cell gets activated, so right now this B cell is not activated. It's just floating through your lymphatic tissues or your blood, just having a boring day. If it binds to the receptor, it activates, all right? I'm sorry, if it binds to the antigen, it activates. And when it activates, what it does is it starts secreting antibodies. And an antibody is basically just this same receptor, but it gives it off. So antigen receptors and antibodies are the same thing, except antibodies are free floating in the blood. Antigen receptors are attached to a B cell or a T cell, okay? This word immunoglobulin is just what you call the antibodies that are just flowing through your blood, all right? This point here, antibodies are like the receptors, but they're not membrane bound. And what's unique here, or what's important here, is that the B cells themselves don't fight off pathogens. B cells produce antibodies, and the antibodies are what defend against pathogens, okay? If you need to go back and watch the last 5 minutes again, do that because it's about to get even more complicated. So this just shows, so I have a B cell, it attached to an antigen. Here's the epitope where it actually attaches. It activates, it gives off the antibodies, which would go out and attach to other antigens or other types of the same antigen. So now let's transition to T cells. So T cells are similar to B cells, but there are some, it's a little bit more of a complicated story. So T cells, again, remember they have, they're made in the thymus on the heart. They have these sort of eye-shaped antigen receptors. There's a variable region, there's a constant region. It's not that different from B cells. The difference, though, is that T cells, let me skip ahead to this diagram. I'll go back to the notes in a minute. T cells respond to the antigen in a somewhat different way. So look at this picture. Here I have some kind of phagocytotic white blood cell that engulfs the pathogen. So this green thing, this is called a major histocompatibility complex, an MHC molecule. These are proteins that are made by your cells. They're your proteins. And what they do is they basically, this white blood cell engulfs a pathogen, breaks it down, and it takes a piece of the antigen. It's almost like a trophy. And it binds to this MHC molecule and moves it to the cell membrane, and it presents it to a T cell, okay? So T cells are activated, a little bit different from B cells. T cells are activated by an MHC molecule, which are yours, that's presenting a piece of the antigen, kind of like a trophy, to a T cell, okay? So going back, so like I said, T cells only bind to antigen fragments, which are displayed by an MHC protein, okay? So we've already kind of gone through this slide. So if infected cells, they break down the antigen into fragments. What's necessary is that you have to have the interaction between the T cell and the antigen fragment on the MHC molecule. So all of this that's in the notes here, this diagram is showing it. You need to have the MHC molecule and you need to have the antigen being presented to a T cell to activate the T cell. Okay, so the way that B cells and T cells develop, there's four main sort of general characteristics. So there's a diversity of lymphocytes and receptors. There's millions of different types of T cells and B cells. Cell tolerance means that your B cells and T cells generally do not react to your own cells or the molecules on your own cells. Once B cells and T cells are activated, they proliferate rapidly. And then you have what's called immunological memory, which is basically if you see an infection a second time, your body responds to it much faster than it did the first time. So let's go through these quickly. So, you know, with all the different variable elements that you have for the antigen receptors, your immune system can respond to millions of different types of infections. If you look at the different genes, what these Ig genes, the Ig again stands for immunoglobulin. If you look at the numbers, I don't care about the numbers, but if you look at, like, this is an example of an undifferentiated B cell. We've discussed the whole idea of introns and exons, how you can cut out the introns so you only have the exons, and how you splice genes different ways can give you multiple different versions of a given gene from one parent gene. So basically, you know, you don't have millions of genes for immunoglobulins. You have a set number of genes, but you can splice them different ways and arrange the variable regions and the constant regions so that you can have millions of types of B cells and T cells. The slide that I just skipped, again, don't worry about numbers, just recognize that through alternative gene splicing, you can have way more combinations than you do original parent genes. So the general way that the story works, so your lymph nodes, you have lymph nodes, you know, in your armpit, your neck, and your groin. They're all over your body, and you have lymphatic vessels. So these B cells and T cells sort of tend to hang out in your lymph nodes, and, you know, they're exposed as a foreign antigen is going through your body. It's exposed to countless T cells and B cells. Most of them will not bond. All right. But once you have a B cell or T cell that does bind to an antigen successfully, they get activated and they go through a process called clonal selection, which is basically they go through mitosis a gazillion times to where one B cell that gets activated becomes millions of B cells very, very rapidly. All right. There's two kinds of B cell or T cell clones. There's what are called effector cells, which are very short-lived. Those are the ones that, you know, in the short time, take care of the pathogen. Then you have longer-lived, what are called memory cells, which the body hangs on to for decades. And if that same antigen comes back again, you know, you get chickenpox or you get the chickenpox vaccine, Once you're now immune to it, if your body sees that antigen again, it responds to it so fast that you never actually get sick. Memory cells can last for decades. You know, there are cases like shingles. You might get shingles when you're older. Shingles is the same virus as chickenpox, but your memory cells, they don't last forever. After decades, your immunological memory might start to fade, such that old viruses can infect you again because all your memory cells are gone. But again, they last for decades. So the idea of clonal selection, like I said, so here I have an antigen that has this weird, half of it's round, half of it's kind of pointy. I have 3 different kinds of B cells. The one that binds is the one that has the purple antigen receptors. So this one gets activated, forms millions of copies. Some of them become memory cells, which just hang out until the antigen comes back. Then you get what are called plasma cells or effector cells. Those are the ones that actually give off the antibodies, where the antigen receptors are released, and the antibodies are what's going to go out and actually handle the pathogen. So immunological memory. So I said this just a minute ago. So the first is exposure to a new antigen. It might take, a couple days, it might take a week or more to get a B cell and a T cell response that's sufficient to fight it off. The secondary immune response, because you have those memory cells, is so much faster. And again, it can last for decades. This is a very common graph showing this. So the x-axis is time in days. The y-axis is antibody concentration. You can measure an immune response by how many antibodies you're producing. So I was exposed to antigen A around day, I don't know, like 5 or 6. And it took about a week for me to build antibodies to A. Then the antibodies go down because I'm well, I fought it off. Around day 28, you're exposed to antigen A again, but look how quickly you make antibodies. You don't even get sick. And actually the graph shows I was first exposed to antigen A around day like 2, where this arrow is. Around the same time, actually on the same day, you're also exposed to antigen B. Antigen B you haven't seen before, so it goes through the same type primary curve as you had when you first saw antigen A, okay? If you're exposed to antigen B again around day, 70, you would get a massive immune response such that you probably never even got sick, all right? And again, usually when you're sick, the symptoms of being sick, a fever, a sore throat, runny nose, that's not from the antigen, that's from your body fighting off the antigen. So the 2 branches of the adaptive immune system are called the humoral immune response and the cell-mediated immune response. So humors is kind of an old-fashioned term for body fluids, like the humors of your body or your blood, your lymphatic tissue, your bile, whatever. The old days of the 4 body humors. The humoral immune response talks about the immune response that relies upon antibodies, which come from B cells. Right, so anything that involves B cells is the humoral immune response. And again, those antibodies are carried in like your blood and your lymphatic tissue, so that makes sense to call it the humoral immune response. The cell-mediated immune response relies upon T cells. It relies upon cells, so it's called cell-mediated to destroy the pathogens. It's a little misleading because T cells are also in your body's humors. So this isn't, the terms aren't necessarily the best terms, but that's what we use. Humoral involves B cells. Cell-mediated involves T cells. Humoral relies upon antibodies. Cell-mediated relies upon specifically T cells. So let's go through these. So types of T cells. So helper T cells, that's a great name, are cells that help out, all right? They boost what's already going on. Helper T cells can simulate both the humoral response and the cell-mediated response. And going back to what we said before, so here I have the general idea where I had an antigen, it got engulfed by some kind of white blood cell. You presented the fragment in a MHC molecule, and it presented it to a helper T cell. Helper T cells don't fight off anything. Helper T cells produce cytokines. They produce chemicals that cause, in this case, B cells, or what are called cytotoxic T cells to divide rapidly. all right? So helper T cells help with clonal selection, and that they stimulate the correct cells, in this case, the correct B cells or what are called cytotoxic T cells to divide. And helper T cells are activated by this MHC complex with the antigen. And remember, antigen presenting cells are the ones that have, there's a class 1 and class 2, don't worry about that, have the major histocompatibility complex, the MHC molecules on their surfaces, where they take the antigen as a trophy and present it, in this case, to a helper T cell. All right, so helper T cells trigger both the humoral and the cell-mediated response. Cells called cytotoxic T cells, the name kind of gives it away. These are the types of T cells that actually fight off invaders. Oftentimes they will contain proteins, or they'll secrete proteins that disrupt the membranes of the pathogen and kill them. Here I have a cytotoxic T cell that's been activated by binding to the MHC antigen complex that's being presented by a cell that's infected. And the cytotoxic T cell gives off chemicals that cause the infected cell to die. So basically, this is your body killing its own cells, but cells that were infected with the pathogen. The name gives it away. Things that are cytotoxic, guess what they do? They kill cells. So B cells and antibodies. So B cells are also simulated by helper T cells to go through clonal selection. So if you look at this diagram here, in the first box, I have a white blood cell that engulfed the pathogen and produced the MHC antigen fragment complex, and it's binding to a helper T cell, which is now activated. This helper T cell goes and attaches to a B cell that has the same antigen. That B cell, or the helper T cell, produces cytokines, which cause that B cell to go through clonal selection, and it forms two kinds of B cells: memory B cells, which will be used if that same antigen comes back again in a later infection, and then plasma cells, which are the ones, again, they give off the antigen receptors, which at this point we call antibodies, and the antibodies will then go bind to the pathogen and take care of it. So in both cases, this slide and the previous slide, what activated either B cells or cytotoxic T cells was a helper T cell that had been activated by binding to the MHC complex with the antigen and giving off cytokines. So on this slide, we discuss how the antibodies themselves don't actually kill the pathogen. Antibodies, through various means, sort of target the pathogen for destruction. In some cases, antibodies will bind, they'll kind of clump together and they'll bind to a bunch of the pathogen and cause it to just become a big clump. And if that's a virus, clumping the virus will prevent it from entering cells, so you've basically neutralized it. Sometimes they'll bind to toxins, and if a toxin can't get into a cell, you've kind of neutralized it. In some cases, they'll activate the complement system, which is that group of 30 proteins that help target pathogens. And the complement system, if it's activated, will cause an infected cell to lyse open, or a foreign cell to lyse open. So again, through various means, antibodies don't actually kill anything. They target them for whatever pathway for destruction, either neutralizing them or causing them to clump together, or targeting them for a white blood cell to come later and take care of them. One thing to point out too, if you have the antibodies for an antigen, it means you either got a vaccine for that antigen or you were infected by it at some point. So like when I was a kid, I got chickenpox. So I have chickenpox antibodies in my blood right now. You probably got the chickenpox vaccine. So you also have antibodies in your blood for chickenpox. One way to test if you're immune to something is you do a blood test called a titer, and it's a test that tests for the antibodies, right? So if if they do a blood test and if I have antibodies for COVID-19, it means that I had COVID-19, all right? Why would you have the antibodies unless you were infected previously, or if you got the vaccine, if there is a vaccine? So this slide shows this diagram, which actually I want to skip ahead to the next slide because it shows the diagram bigger, that summarizes the entire adaptive immune response. This seems complicated, and it is, but let's just go through this because it explains everything. So you're exposed to an antigen, right, one you haven't seen before. It's engulfed by some kind of white blood cell that breaks it down and presents the epitope, a piece of the antigen with an MHC protein complex, which are in your cells. on the surface of its cell membrane and presents it to a helper T cell. The helper T cell gives off cytokines that activate B cells and cytotoxic T cells. The antigen itself can also activate B cells directly. And the antigen presenting cell can also activate cytotoxic T cells directly, okay? The AP exam wouldn't expect you to have this memorized, but if they gave you this diagram, you should be able to write, 10 pages on this diagram. So again, helper T cells give off the cytokines to produce B cells in the cytotoxic T cells. B cells then differentiate into plasma cells, which give off antibodies, and memory B cells for a later infection. Memory B cells later can become plasma cells to give off antibodies, again, if you're infected with the same antigen, a year later, 10 years later, sometime in the future. Helper T cells also produce what are called memory T cells. Memory T cells can become cytotoxic memory, or they can become memory cytotoxic T cells, which can become active cytotoxic T cells upon a later exposure. All right. Notice in this diagram, what's in the center are helper T cells. Without helper T cells, will you get an immune response? Yes. If I took this whole thing out, there's still pathways of getting to the effector cells, right? But the helper T cells, are the gas pedal for your immune response. If you take them out, your immune response, you might have one, but it's so slow that you can't effectively fight off, the flu or a cold or pneumonia or really any kind of infection. This is going to be an important concept later. So you might want to pause the video for a second. So just take some time with this diagram. This diagram summarizes the last 10 minutes of the video. And if you understand what's going on in this diagram, then you understand the immune system. So active and passive immunization. So we're going to discuss vaccines in a minute. Active immunity, when you are exposed to a pathogen and it infects you and you get over it and now you're immune to it, that's called active immunity. Passive immunity would be, an example is when a mother breastfeeds a baby, antibodies from her blood actually wind up in breast milk. So if the mom has antibodies against chickenpox, some of those antibodies will wind up in the baby, and as long as the baby's breastfeeding, it's going to be immune to chickenpox. The difference here is it's short-lived because the baby can't make the antibodies itself. You're giving it the antibodies, which will work until those antibodies go away, and then it can't make its own because it doesn't have the memory cells. And again, the way vaccines work, vaccines trigger an active immune response by introducing, usually a vaccine might be a piece of a virus protein code or a piece of a bacterial cell membrane, something that sort of tricks the immune system into becoming immune to a part of the antigen. That part doesn't make you sick because it's only part of it. But if you later saw the full antigen, you would fight it off because you're already immune to it. Just really quickly here, monoclonal antibodies. So you can use antibodies as tools in science. And basically, if I can get a B cell that say is activated against COVID-19, and I can grow that B cell in culture in a test tube or a petri dish, and I can sort of harvest the antibodies from it, you could use that in medicine. That term is called monoclonal antibodies because you're growing antibodies of just one variety, which is useful sometimes in medicine. Immune rejection. So, if I give you a kidney transplant and it's from a donor where those MHC molecules we keep discussing are different than yours enough to where you would have an immune response to that kidney and you would reject the kidney, that would be bad. So usually when you do organ donation, you want to find a match, and a match just means you have similar MHC molecules in your blood cells. And even if it is a decent match, you still usually give the recipient of that kidney medications that suppress their own immune system, at least for a while, to prevent rejection. Of course, if you're taking drugs that suppress your immune system and you get the cold, you're going to get very sick because you can't fight off the cold. If you got pneumonia, you could potentially die on those medications. So usually you're in isolation for a period of time after you've received an organ transplant, because you want to prevent rejection, but you also don't want to weaken the person so much that, you know, a cold is a life-threatening condition. Allergies, we discussed this already. Allergies are when your immune system is being hypersensitive to things like pollen. They might trigger that inflammatory response with histamine. Pollen should not trigger an immune response, but it does, because our immune systems can go overdrive sometimes. And histamines, again, are medications like Sudafed that block the receptors for histamine, so it diminishes the immune response. This just shows what's going on here with these mast cells. Here we have pollen granules, which when they bind to the receptors of mast cells, the histamine gets given off, and the blood cells or the blood vessels in that localized area become dilated, and you get the inflammatory response. The extreme version of that is called anaphylactic shock. So, you know, things like bee venom, peanuts, shellfish, if I have an intense immune reaction like around my airways, and they like the vessels dilate so much that my air passageways get blocked off, you can actually suffocate. That's called anaphylactic shock. Things that the drug epinephrine, like an EpiPen, is, you know, things that people that have hyperallergies might carry with them, because it counteracts the immune response. It counteracts the inflammatory response very, very quickly to prevent your airways from closing up. Autoimmune diseases, things like, this is a picture of someone that has rheumatoid arthritis, and basically that's a condition where your immune system attacks the cartilage between your joints, which obviously it should not do. The form of diabetes, I guess it's type 1 diabetes, where your immune system attacks your insulin. Multiple sclerosis, these are all diseases, lupus, where your immune system is attacking your own cells, which, you know, you don't want it to do that, right? The way that pathogens avoid your immune system, so remember the word epitope. The epitope is the part of the antigen that the white blood cell responds to. If the virus or the bacteria mutates and changes that part of the epitope, suddenly you aren't immune to it anymore. Because this, your immune system works upon physical, like touching recognition. If the part of the antigen that the white blood cell touches changes, then you're not immune to it anymore. And viruses, they mutate. Some viruses can become latent. If a cell is infected with a virus, but the virus doesn't actually divide and leave the cell, your body has a hard way of knowing that it's infected. Viruses like herpes can lay dormant for a while, and again, if they're dormant, if they're not dividing, they're hiding from your immune system. They would become active again when your body's under stress, you have a fever, a woman's menstruating, when there's other things going on that are distracting your immune system, the virus might become active again because it knows your immune system is suppressed. So AIDS, so we've discussed AIDS before. AIDS, the HIV virus, remember I said to note where helper T cells were in that diagram? The AIDS virus, or the HIV virus, infects helper T cells. And remember, the HIV virus can be dormant for like a decade. So after a decade of it being latent, basically all of your helper T cells are infected. And if the virus becomes active at once, then your helper T cells are just gone. And people that have AIDS, which stands for acquired immunodeficiency syndrome, you don't die of AIDS. You die because you get pneumonia and you can't fight off pneumonia, and pneumonia kills you, or the flu kills you. Having a cold, if you have AIDS, is a very, very serious condition because your immune system is just wiped out because you have virtually no immune response to anything. If you have HIV, any kind of infection is very serious. There is no cure. There are drugs that are antiviral drugs that will slow down AIDS or the HIV virus, such that as long as you take the medications, you're not going to die. But if you stop taking them, you're going to get the AIDS will flare up again, and you're going to eventually die from it. So last thing, so cancer and immunity. So this says 20% of all cancers in humans are caused by viruses. So here's the deal. We've discussed this before. Viruses can insert themselves into your genome. And if a virus disrupts a proto-oncogene, that proto-oncogene is now an oncogene, and those cells are now cancerous. Viruses can cause cancer because they mess with your DNA. Cancer always has some kind of genetic root. It could be a mutation in the cell itself due to a sunburn, or it could be a mutation because a cancer gene, disrupted a normal gene that controlled the cell cycle. Maybe the viral gene got right in the middle of a tumor suppressor gene. That tumor suppressor gene doesn't work anymore, so you don't suppress tumors. Suddenly you have cancer. Sometimes viruses could accidentally have a proto-oncogene from someone else in it, and now you have extra proto-oncogenes, and if you have extra ones, they're going to be overexpressed. Their oncogenes, the cell cycle gets the gas pedal gets pushed down. We've discussed before HPV, which is a very, very, very common STD. Most people have HPV. HPV is not especially harmful, the virus itself, but HPV causes over 90% of all cervical cancers. In 2006, it's the Gardasil vaccine. A lot of you might have gotten it. Usually you get it when you're like 12 or 13. The Gardasil vaccine is a vaccine against HPV. If you can't get HPV, you have a basically minuscule chance of getting cervical cancer, because most cervical cancers are caused by HPV. Guys can't get cervical cancer because guys don't have a cervix. So usually you don't give the Gardasil vaccine to guys, but HPV can cause different kinds of throat cancers in guys. It's just, one of the deadliest cancers in women is cervical cancer. So usually we don't give it to guys, we give it to girls. But giving it to the entire population would wipe out HPV, which means you would wipe out cervical cancer. Okay, that was a lot for one chapter. That's definitely enough. I hope that was helpful, and I will see you guys next time.